Topic 25b, Agents of Evolutionary Change. In the last video, we used the Hardy-Weinberg formula to calculate allele and phenotype frequencies within a population. And we used the Sesame Street Muppet aliens as an example. Biological evolution occurs when the frequency of dominant to recessive alleles changes. In this case, it would change from a 1-to-1 ratio to something else. And when the allele frequency changes, the phenotype frequencies will also change. So what can change genetic diversity, also known as allele frequencies, within a population? We will consider 5 different things: mutation, gene flow, non-random mating, genetic drift, and selection. So let's explore each one. You may recall that mutation is a change in the base composition of DNA. Some of these changes can change protein composition and subsequently the phenotype of an individual. One way mutation can change genetic diversity is by creating new phenotypes. The second one, gene flow, is the movement of alleles from one population to another. One simple example of gene flow is to imagine 2 populations of a species separated by a physical barrier, or living on 2 islands. Each population has a unique allele frequency that is maintained as long as they are separated. If the physical barrier is removed, say by a land bridge between the 2 islands, then the 2 populations can become a single population, and the allele frequencies will also change. The third phenomenon, non-random mating, is when individuals with a certain genotype may only mate with other individuals with a certain genotype. So if we go back to our islands, the allele and genotype frequencies shown when the 2 populations intermingled assumes random mating within that single population. If there was non-random mating, that might mean that only the reds mate with other reds, or only the blues mate with other blues. So they could essentially maintain 2 separate populations even though they are in contact with one another. So there are 2 types of non-random mating. Assortative mating is where mates are similar to one another, phenotypically similar. Disassortative mating is when mating pairs are usually phenotypically different. Neither of these are random mating because they show an aspect of choice by the mating pairs. The fourth phenomenon is called genetic drift, which is a phenomenon of small populations where the frequencies of particular alleles may change drastically by chance alone. An example of genetic drift could be a catastrophe like a natural disaster that kills a portion of the population. In this case, we'll say it's a hurricane that randomly removes the majority of the red individuals. When this occurred, the allele frequency was shifted to favor blue. So most of the offspring of this population would be blue, even though the red allele is dominant. There are 2 types of genetic drift. The founder effect is when a small group of individuals migrate away from the main population and, say, form their own colony with no contact with the original population. And there's also the bottleneck effect. This is when a natural disaster occurs or disease drastically reduces the size of the population. The 5th phenomenon, selection, will be covered in the next video. To quickly review, there are 4 phenomena covered in this video that can change allele frequencies: mutation, gene flow, non-random mating, and genetic drift.